So, most welcome, everyone, to the Royal Academy of Engineering Sciences, or EVA, as we say here in Sweden. We will listen to the seminar of today, Genome Editing, to Feed, to Fuel, and to Cure, which of course refers to the versatility of the CRISPR-Cas9 technology. And I'm super excited about this seminar. And I'm really happy to see such a crowded house. We haven't had such a crowd since the former Prime Minister of France was here. It's true. So either it's a very exciting subject or it has to do with France. That we don't know. My name is Stefan Ståhl, and I'm the chairman of Section 10, Life Sciences of EVA. And I've had the pleasure to put together this seminar with the help of my co-pilot, Lena Gustafsson. And it's indeed a true pleasure to have, an honor to have Professor Emmanuelle Charpentier here as keynote speaker, presenting her CRISPR-Cas9 case to us. It's fantastic. There is no way that I could use my few minutes here to present Emmanuelle in a proper way. Just to go through your awards and honors over the last couple of years would take the full afternoon, so we cannot, unfortunately, do that. You have to trust me on that, though. We have also two very exciting additional speakers, Stefan Jansson and Paul Hudson, who will share their applications of the technology with us. And we have Jennifer Kahn, who will give an introductory remark and also act as moderator for this. She is from sunny California and took her all this way over to do this for us. Jennifer is a science journalist, giving an extra perspective to this, and a true knowledgeable person in CRISPR-Cas technology. And your 2016 TED Talk on the subject has been viewed of more than 1.4 million viewers. So you're very well suited to act as moderator for this seminar. I have to go through some rules. And first of all, this seminar is live streamed. So welcome also those that look from other places. And it will be made available also 4 weeks after this seminar. For that reason, when you ask questions, you have to use the microphones that are mounted on the side of your chairs. And if you don't happen to have a microphone, borrow your neighbor's. And the questions for each speaker, we keep them at the end of each seminar to help Jennifer and us all to keep time. And then, if everything works well, we will have a time for a panel discussion at the end when all of you are allowed to ask questions to all speakers at the same time. We have to be ready just after 7 because registered guests will have dinner at 15 minutes past 7. And I promised to get that right, so otherwise I'm done. So by that, I leave the floor to Jennifer, and most welcome. Thank you.
Thank you so much, Stefan. Well, as Stefan said, my job in these first 10 minutes is to give you an overview, kind of the ordinary person's overview of the gene editing technology CRISPR. And since we are lucky enough to have the co-creator of CRISPR here with us, Dr. Emmanuelle Charpentier, I'm not going to talk about how the technology was invented. I'll leave her to do that. Instead, I'll just explain briefly what makes CRISPR different from previous gene editing technologies, what we can do with it, and what questions and challenges we might be facing given these new abilities. All right, part 1. What makes CRISPR different from previous gene editing technologies? Basically, the most important thing to know about CRISPR that when it was invented just about 5 or 6 years ago, it outperformed pretty much every previous gene editing technology by a long shot. It was faster, it was cheaper, it was easier to use, and it was more precise. So to give you just a couple examples, before CRISPR, it would take a graduate student roughly 2 years to engineer a mouse with a single genetic mutation. With CRISPR, it can take as little as 2 days. On top of that, you can use it not only to replace one gene with another, but even this level of precision to alter a single base pair within a gene. So it's almost like having a word processor for gene editing. Another thing that made CRISPR revolutionary was that it worked in almost every animal, from silkworms to monkeys, and also in almost every cell type. Kidney cells, heart cells, you name it. This was a big deal because a lot of our old gene editing techniques simply didn't work in a lot of stuff. You could genetically engineer mice, but even rats were very hard to do. Finally, one last thing that CRISPR changed was how many genes we could edit at once. So before CRISPR, scientists could edit basically a single gene at a time. And with CRISPR, we can now edit multiple genes simultaneously. And this is important because most diseases are not caused by a single mutation, but by many different genes in combination. Okay, so that's a brief summary of what makes CRISPR different and why it's so powerful, which brings us to part 2 of this little overview, which is what new things this technology is going to allow us to do. So first, I should point out that CRISPR is still comparatively new by the standards of scientific developments. It's only been around for these 5 or 6 years. That said, even in this short time, the promise of the technology it's really been borne out. It's revolutionized almost everything. I think it's actually fair to say that literally any industry that involves genes has been affected by CRISPR or any line of research. That includes things like biofuels, which Dr. Hudson is going to be talking about a little later this session. But it also includes things like yogurt and cheese, which depend on bacteria. And in fact, that was one of the early applications of CRISPR, was to create healthier bacterial cultures for yogurt. It's also used even to create dish soaps and laundry detergents because those rely on enzymes to break down grease or to make clothes soft. So it's kind of everywhere. But the 2 places where CRISPR is probably likely to have the biggest impact are agriculture and medicine, at least so far as we can predict. So in agriculture, one problem that researchers are kind of frantically trying to solve is how will we feed 10 billion people by 2050, using roughly the same amount of land as we do now. And one example might be to genetically engineer cassava, which is a core food for about 800 million people, but which contains cyanide and has to be sort of carefully processed to be edible. And this is a big issue, especially in a lot of African countries. So using CRISPR, scientists have figured out how to silence the cassava genes responsible for this cyanide production. And they also have found ways to do things like create CRISPRized pigs that are resistant to swine fever, which is a disease that's really been ruinous for farmers in sub-Saharan Africa. So I will say that this is the same kind of thing we've been able to do in the past with older gene editing techniques, but CRISPR now allows us to make these changes much more quickly and with greater precision, which is important, especially because it's very likely that we will need to do things like make crops that are more drought resistant or more tolerant of hot weather, especially as climate change continues. So we'll likely be coming to rely on this more and more. And of course, these developments are also going to have policy implications. And Dr. Janssen will be discussing the very interesting question of whether crops and livestock that have been edited with CRISPR will or should be still labeled as GMOs. All right, so that's a quick overview of some of the agriculture applications. Very briefly in medicine, CRISPR has opened the door to a number of things. One of these which has gotten the most media coverage probably is that it's led to new gene therapy treatments for a range of monogenic diseases, including, so that means just a single gene that has a mutation. And that includes sickle cell anemia, beta thalassemia, that's a blood disorder, retinal dystrophy, which causes blindness. I think Dr. Charpentier may discuss some of these. But even beyond this sort of treatment, CRISPR has radically accelerated our ability to study the underlying genetics of disease. So for instance, until recently, it was essentially impossible to do genetic engineering in T cells, which are a crucial part of our immune response. And with CRISPR, we can do that. And that means we might finally be able to get a handle on these elusive conditions like rheumatoid arthritis or diabetes. And it also means that we may finally be able to begin to explore the genetic roots of complex mental disorders like autism and schizophrenia, which are thought to be caused by the interplay of dozens of genes. Okay, finally, one of CRISPR's biggest impacts will likely be in drug development, and that's because pharmaceuticals are often designed to correct for something a gene is doing wrong, like making a protein that raises your cholesterol. And the hard part has always been figuring out which genes matter the most to a particular disease. So to do that, researchers typically conduct what's called a genome-wide association study. And that means they sequence the genomes of a big batch of people who have a disease and then look to see which of those genetic variations are shared between people. And that's a great approach, but the problem is there's so much natural genetic variation in people that's really hard to tell sort of the signal from the noise, to tell which of the hundreds of overlapping mutations actually matter, which are the important ones. And so before CRISPR, it would have taken just decades to genetically engineer and test each one of these little variants to see what effect they had. But now using CRISPR, we can relatively quickly figure out what each variant does and which ones therefore might be important targets for medication. All right, so that's your very, very brief, high-level overview of some things CRISPR has allowed us to do. And it's going to just bring us to the last part of this talk, which is some new questions and challenges this technology is probably going to raise. So in terms of what questions it's going to raise, the answer is obviously quite complicated, given all the ways that CRISPR can be applied in the world. But I want to address just a couple key things. So first, when it comes to the use of CRISPR on people, one of the main things that's gotten a lot of media attention has been this thing called germline editing, where scientists edit the DNA of an embryo in a way that doesn't make a change just in that individual, but then is heritable. It can be inherited by the children and the grandchildren. And the concern about this in humans is that, or one of the concerns in humans is that the wealthy would be able to pay to, say, increase, their chances of their child being very attractive or being more intelligent, or just simply to reduce their risk of developing Alzheimer's. And this is a very interesting ethical question to consider. But I do want to be clear that in practice, it's probably still quite far off. So far, scientists have successfully edited a human embryo to remove a disease-causing mutation, which is great, but no edited embryo has yet been brought to term, and the idea of optimizing an embryo for something as complicated as intelligence is still quite far out, partly because we don't understand all the genetic factors contributing to intelligence, but also because we don't know what else will happen if we change this piece of a gene, right? There may be unintended consequences or crucial other processes that might just collapse. So this is probably not something we're going to see in the next few years. You never know the pace of technology today, but let's say probably. Beyond that, there's one last application of CRISPR that I especially want to talk about. It's a particular interest of mine, and it's different from what anyone else is going to speak on, and that's something called a gene drive. So some of you will have heard of gene drives, but for those who haven't, a gene drive is a tool that drives a genetic change through a population. So if you have a bunch of mosquitoes with white eyes, that's their normal color, and you want all their offspring to have red eyes instead, you can use a gene drive to just force that change into subsequent generations so that after, 5 or 6, whatever it takes, all the mosquitoes will have red eyes. So researchers had actually been working to create a gene drive for about 2 decades, but the ones they'd made were extremely weak. They increased the chance that a particular trait would get inherited by about 1%. And then in 2015, just very recently, a graduate student figured out how to use CRISPR to make a gene drive that was literally 100 times more powerful. It basically guaranteed that a trait would get inherited 95, 98% of the time, at least in the lab. And so it's, and that, again, not just inherited once, but inherited until it really spreads through the population. Okay, so why does this matter? Why am I telling you about all this? Well, the good news is that it opens the door to some potentially remarkable things. So right now, there are 2 main types of gene drive. There's something called a replacement drive, which spreads a small but important trait, like making a mosquito that can't transmit malaria, which is probably going to be one of the first applications of gene drive, or fruit flies that don't lay their eggs inside cherries, which would be a great help to those who are working with invasive pests in agriculture. There's also something called an extinction drive, which would cause a species to produce only, for instance, male offspring, which would eventually, after enough generations, result in a population drop. All men, you don't have a lot of reproduction. So if you put an anti-malarial replacement drive in Anopheles mosquitoes, which is the main species that transmits malaria in Africa, it could spread through the population of wild mosquitoes, which has always been the problem. We can make these mosquitoes in the lab, but then how do you spread them out? And that in turn you could make these, spread this anti-malarial capacity in the wild mosquitoes, and you could see a dramatic reduction in the number of new malaria infections. So a lot of foundations, including Target Malaria, are extremely excited about this. It's just another way to mitigate this disease. All right. People have also talked about using extinction drives to get rid of, for instance, invasive species. So you could use them to remove rats from the Galapagos Islands without using poison, or get Asian carp out of European rivers. So in theory, this is great. It means we could restore hundreds of native species that have been kind of pushed to the brink already. And it could also potentially do a lot for agriculture by eliminating pests, like that fruit fly I mentioned that lays its eggs inside cherries, or something like the diamondback moth. All that said, it's not yet clear exactly how well gene drives will work in the wild, and researchers are also still evaluating the potential risks. So in the case of malaria, the argument for not using a gene drive would have to be quite strong, because the status quo is that the disease kills several 100,000 people a year. So in order to be worse than that, it has to be pretty bad. What's a little less clear is whether a gene drive will end up making sense for invasive species or agriculture. What we don't want is for a dozen Asian carp with an all-male gene drive to accidentally make their way from Europe back to Asia, where they could potentially wipe out the native Asian carp population, right? But what scientists are working on right now is, hopefully developing gene drives that don't have that risk, either because they, for instance, target a gene that specifically affects these new invasive European carp, or because the gene is designed to be self-limiting. The drive is designed to be self-limiting. Okay, and then this is where the governments and NGOs going forward will have a vital role to play by creating intelligent regulation that can weigh the risks and benefits of gene drives on a case-by-case basis. And just this past July, the Netherlands passed an amendment that requires all applications for gene drives to undergo a risk assessment that they've developed, but which will also permit the technique to be used should the risks for humans and the environment turn out to be, relatively negligible. So I'm almost out of time. This is a very brief overview. But as a very final point, I just want to note that both this idea of a CRISPR-based gene drive and CRISPR itself, they're not static technologies, right? So they're going to evolve and change all the time. And just to give you a brief example, researchers recently created a new version of CRISPR that targets RNA rather than DNA. And that's big news because many diseases, including Huntington's disease and ALS, are caused not by a mutation in our DNA, but by a typo in our RNA. And other researchers also recently created a different version of CRISPR that doesn't cut and paste genes, but simply turns them on and off using chemical signals. And this matters because it means that we can now do something called epigenetic editing, as well as ordinary gene editing. So it's basically giving a volume control knob for how much protein a gene produces. And that's important because in some diseases, the problem isn't that the gene is producing the wrong protein. It's that it's producing too much or too little of it. And already in mice, at least, this technique has been used to cure both diabetes and muscular dystrophy. In mice, but still, it's good news. So to sum up, basically, CRISPR is a sort of an extraordinary new tool. And like all extraordinary tools, will have benefits and some other risks. And going forward, our most important job is to really thoughtfully consider those and to weigh them with intelligence and candor, honestly, to have an open discussion and let the facts speak for themselves. So next, we have Dr. Stefan Jansson, who will be talking about CRISPR editing in plants and European GMO regulation.
Okay. Thank you very much. And do I have to press something or not? Oh, no, that was not me. No, so I will not talk very much about technique here. It would rather be how this could be used in agriculture, but more or less whether it will be possible or allowed to use it in agriculture. Should be on. Okay. Do you hear me now?
Yeah, better.
Okay, good. So, but I will start actually talking just about plant breeding in general, not mentioning CRISPR-Cas in the beginning. And I sort of like this picture. This really shows how plant breeding has been done for millennia. So this is cabbage, wild cabbage growing in the Mediterranean area. It's been growing there for millions of years. And then, of course, through the spread of agriculture back 1,000 years ago, cabbage was starting to be grown throughout Europe. And somewhere, at one point, we don't know where and when, one farmer found one individual plant that had a mutation in a gene that was necessary for the extension of the branches. It was supposed to be a branch with leaves on, but then if there was a necessary gene for that was mutated, the branches did not develop. The branches were of this length, so they had all the leaves on the same spot. That particular farmer somewhere, he thought that either he thought it looked funny or it tasted good, so then he kept it and kept the seeds from this one. And now we eat the offsprings of these ones as Brussels sprouts. Some other farmer somewhere did the same with a plant where there was a mutation in the gene that was necessary actually to form the whole inflorescence with flowers. And then that particular plant developed a structure. It's more or less like a tumor on the top of the plant, which was whitish, and he thought that tasted good, and then he kept that. Kohlrabi, that's a mutation in a gene necessary for the elongation of the stem, and so on. All these things, that's the same plant species with that individual mutation. Of course, those that developed those or identified those in the old days, they didn't have a word for plant breeding. There was no word for plant breeding. They were just keeping the seeds from year to year. And this is how mankind have been developing plants that we eat today for thousands of years in a quite an efficient way, as you see here. And then you can start crossing them with each other and getting new varieties. This was how things worked up to 100 years ago. 150 years ago, we can start, then we had the Mendel sorting out the laws of genetics, and then this process could become science-based, becomes much more efficient. After a couple of decades, the breeders realized that they sort of ran out of the natural variation that was out there. Of course, they had these guys, they could cross them to each other, but they realized if we can increase the variation in the germplasm, then we could create plants that would be even more useful. Then scientists started to expose the seeds to different kinds of treatments that were inducing mutations in them. Either it was radioactivity. This is the Institute of Radiation Breeding in Japan, where they take seeds and just pumps a lot of radioactivity to it. Radioactivity, as you know, induces mutations. 99% of these ones, they will be very bad. They will never be used in agriculture, but from time to time, there is something that is useful for us. Then it can be kept, and then we put in the breeding program. Another way of doing it is to add EMS, for instance, or other chemicals that are, for us, carcinogenic. They cause mutations, they cause cancer with us. In plants, they don't cause cancer, but they cause also random mutations. And a lot of the food that we eat today, I would say the majority of the food that we eat today, they are the results of these mutations programs that's been done in the last century. All wheat varieties that we eat today, they are for very ambitious mutations programs because they were introducing different kinds of resistance into the germplasm and so on. So this is what we eat. And all these techniques, they were, when the European or the GMO legislation in the whole world, when that was formulated something like 30 years ago, there was this belief that everything that we have been doing to produce all the varieties that we eat today, that was regarded as harmless. We don't need to regulate them. So all these techniques, they are without any kind of regulation. When you go in and buy a new type of potatoes or carrots and so, there is no one that ever tried to find out whether there is a danger associated with these ones. In sharp contrast to the techniques that was developed in the 80s, where you were able to move one gene from one individual to another using a bacterium called Agrobacterium. That was regarded to be harmful, or at least there was a danger, because science at that time, we didn't know whether it was harmful or not. Just for the sake of safety, there was a regulation imposed throughout the world saying that these ones have to be taken very careful risk evaluations if they should be used. So it's really an on-off situation. Either you have plants that are not regulated at all, or you have plants that are regulated as GMOs, which in reality means that they cannot be used at all. In particular, not in Europe, because in Europe we don't use them at all. We have a very strict system for that. The reason why there are some very good things to be generated with transgenic technologies is that if you're doing this, you can actually only change one trait at a time. There is if you're growing potatoes in Sweden or potatoes everywhere in the world, you know that the main problem you have is that you may get a disease into your field. It's late blight, it's bladmögel in Swedish, which causes brunröta. This is a disease that has typically very devastating effects on your crop yield. There are natural varieties of potato that has the resistance genes to these ones. The problem is that those natural varieties, they are unhealthy or they are not very productive at all. Scientists have, or breeders have, for about 40 years been trying to cross these natural varieties with the natural genes for resistance into the varieties that we eat today that are healthy and that are producing a lot of offspring. But so far, they haven't gotten there fully. There are varieties around, Charpomira, Charpochona, and so on, which are naturally resistant to this pathogen. But you have probably never heard of them because they are not, I mean, the farmers, they don't want to grow them because they yield too little. So the farmers cannot make a profit out of growing these ones. They can rather make a profit out of growing normal potatoes and spraying them with pesticides to keep away this pest. So of course, you can imagine it would make sense if you could take only the resistance gene from the resistant variety and put it in a potato that is otherwise good. You can get mandel potato with resistance to this. You can get bintje, whatever you want, which is exactly like it is now, plus this resistance. You can never create this with traditional breeding, but you can create it with biotechnology. And this is why this is such an important tool. So, however, if you do this, if you would take this gene from this one and put it into another potato, then it's regarded as a GMO. And then it cannot in reality be used. This is exemplified by this plant here. This is developed in Sweden back in 2005, I think. It was tested in the field in 2006. Fully resistant potatoes developed by the Swedish company. That if that would have been used, could have been all potato growing in Sweden could be completely free of pesticides, because this is the only pest that actually affect our potatoes. But now it's a GMO, which in reality means it cannot be used, and it's still not used and will never be used probably in Europe, because the company that developed this, they believe that they cannot, it's not possible to overcome this blockage of the European GMO legislation. So instead we keep on spraying our potatoes. So the problem here is that although the scientific message is crystal clear, if you compare all these techniques, there are no more risks with these ones than all with the other. We have done enormous amounts of studies on this. Of course, none of these techniques are riskless, but there is nothing that indicates that there is any more risk with these ones than any of the other ones. But we have these 3 letters. It says GMO, and there is a specific legislation for that. That is why people still going around like this and being very worried, very angry, and so on, and in reality blocking these techniques to be used. With that as a background, when the first CRISPR-Cas paper was published, of course, I as a scientist realized the scientific possibilities with this, but also realized the possibilities that this may actually change the whole debate about GMOs. Because now we could start doing some interesting things that I will tell you about in a minute. And I actually started to think, okay, now we have this tool. What should be the first thing that we do in my lab? And after a couple of weeks, I realized that I actually have the example just in front of my eyes, because we've been working, actually I'm a photosynthesis researcher trying to understand how plants harvest sunlight and make them into sugar. This is my basic research project. We've been working with a protein called PSPs, which is a kind of a safety valve. So if there is too much light shining on the plant, it don't want to harvest it all unless it can use it, because otherwise it can produce oxygen radicals. So then they have developed a safety valve that dissipates this heat instead. This is called PSPs. And we had published a paper where we've identified this. We had a mutant, we had actually several mutants, some of them that had been exposed to radiation, so then you had lost a piece of DNA, others that had been exposed to this EMS chemical that has induced point mutations. All of them lacked PSPs, and they couldn't do this process. We published that in 2000. In 2002, we published that if you put these plants in the field, they are less good than the normal ones. This is a good protein for the plants. If you take it away, it will produce less seeds and will be less useful, which made sense, of course, for us. And I've been using this in my lectures, talking about GMOs for years, because I said, okay, if we take away the gene with radiation, it's not the GMO. If we destroy the gene by point mutation, it's not the GMO. But if we take away the same gene using biotechnology, then it's a GMO. For us, they are the same. When we publish them, they are seen as identical. Or you can even put it this way. If we try to publish a mutant like this in a good scientific journal, they will request from us that we confirm it in this way, because these ones are also unsafe. These ones makes a lot of random mutations. So it's impossible to actually ever publish an EMS mutation unless you can prove it with doing a targeted mutation. So for us, these ones are the safe, these are the unsafe, and for the public, it's the other way around, which is kind of an irony. So what we did, we said, okay, we have these ones. Why don't we use CRISPR-Cas to make a plant that is copying this one? So we take away a piece of DNA. That's the only difference from the other one. And we actually did it a little bit tighter than this, because this particular plant, it's not only lacking the PSPs gene, it's lacking a few other genes as well. So now we only took away a little piece of the PSPs gene. And then we went to our authorities, and then we asked, okay, if we do it like this, would this be a GMO or not? And again, it cannot be half GMO. It has either to be a GMO or not a GMO. And what the authorities then had to consider, that was the definition of a GMO in the EU sense, which is written down here. And it doesn't actually say anything about genome editing, of course, because this was made 25 years ago. So now the authorities had to try to make an interpretation. Was this a GMO or not? A year and a half later, they came back and told us, no, if you only take away DNA, if you don't add anything, we don't think this is a GMO. So that was actually the very first plant that we did. So this is lacking this PSPs. And if you make it really complicated, some of the seeds of these ones, they will be GMOs because they will have this Cas9 in them. Others will not have the Cas9, as they will not be GMOs. So I also did a TEDx talk. I only have 32,000, not 1.4 million, but I'm happy with that one. And then I actually did this story, and then, okay, now we have the seeds. And since we have this decision, now in Sweden, we're the only place in the world when we can grow them out in the field. And I was showing the seeds. If you would be a plant scientist, you probably would have noticed that that is not Arabidopsis seeds. They are tiny, tiny. They are other seeds. Because the decision of the Swedish authorities was actually a decision that if you do it like this, you don't even have to ask us, because it's not a GMO. So that meant that every other plant, every other mutation that was done in the same way could be grown in Sweden. So then in my garden outside Umeå, I was growing them, having sort of organic pots and doing it. And for the inviting Gustav Klarin at Vetenskapsradion and at Odla P1 to share with me the first CRISPR dinner in history. Perhaps, because we didn't know at this time whether someone else had done it. So far, no one has stood up and said that they were before. So maybe we were the first one sitting on my balcony there, eating this cabbage. And at the same time, when this went out, it actually made sure that it was Odla med P1 that got the scoop, because he's working there as well. And I was publishing a blog. I was even publishing the recipe, because if now this was the first, we should have it. It was a historical event, so if someone would like to copy it and so on. And Science was writing about it. And this created my 15 minutes of fame. So since then, I've been going around and talking about CRISPR cabbage. So I took this picture when Emmanuelle was in Umeå a year and a half ago, and then she received her 42nd prize. I think now you're up to over 70 or so. At the same day, this was published. My Georgian is not so good. Maybe you know better. I understood that this is probably the journal of the bakers in Georgia, which care about what I was eating for dinner, which I think was fun enough. So I'm sort of becoming the Swedish chef going around. So this is my way into getting famous. But anyway, it became a little bit complicated to go around with the cabbage. So we made a cooking competition at our research center and see if you could make a dish out of this. So this was a winning recipe. So I have a few for some selected guests here can get some chips out of CRISPR cabbage. So there are better chefs than me. I was running a conference last year in Gothenburg. And then after some work, I was able to convince Gustav Trädgård at Sjömagasin, one of the Michelin star restaurants, that they was making a dish out of cod with CRISPR cabbage. So I was saying that now we are in Sweden, now we can use this because this is not regulated. I've been going to other countries because the interesting thing here now, this was the Swedish authorities that was interpreting the European law. So really the implications should be that they'd be the same in the whole Europe. But most other authorities, they didn't dare to take the same step. When I went to Finland some time later, I asked, could I bring the cabbage? They said, no, you cannot bring the whole one, but you can actually bring the shoot if you take away the roots, because then it cannot propagate. So this is me at the Umeå airport taking away before going into Helsinki. A month later, I was invited to Norway. In Norway, they just took in a new food policy in the government. And here they say that, in Norwegian, genetically edited organisms must be regulated through the gene technology law. And they cannot be allowed unless there are guarantees that they are traceable. Now, the interesting thing is, these are not traceable. Because so far, I have never told anyone in public which gene that has been deleted from these ones. And if I don't do that, you cannot find out. That's the way it is. There is, I mean, some of us, Matthias and I, work with genome sequencing. We know in reality, you can only find if you know what to look for, if you add something, but you can never find what is lacking because you can never find anything to 100%. So, Stortinget, they tried to regulate nature, which I think they will have a problem to do. I thought I should eat this when I was passing over the border on the airplane, but then instead I waited until I landed in the airport in Norway. So then I was destroying it by eating, because in Norway they didn't want me to bring it in. But then I kept this little stalk and I was throwing that away in the bin in the airport, in the international area before going through the customs. So then I don't know whether I'm in Norway here. And if someone brings this in for destruction in Norway, so now is he the one that commits a crime in Norway, or am I the one who commits the crime in Norway? You see, it's getting quite ridiculous. So I've been going around now for many countries, and then I always ask the regulators, can I bring a cabbage plant like I would do in Sweden? Or can I shoot like in Finland, or chips like in Norway? But if you say no, if I bring something, if I bring a cabbage there, and I don't tell you whether this is the real mutant or not, what would you do? Because in our European system, I think in all juridic systems, the prerequisite for a prosecutor to press charges against something is that you can provide evidence in the first case that the crime has been committed. If no crime has been committed, there cannot be a charge. So, and in this case, I actually doubt that anyone could ever prove that a crime has been committed to this particular. If the police would come in there, I would say, okay, fine. I was just kidding. all genes are there, and so on, and they cannot prove that I'm wrong. So this has been the situation for some time, up to this summer, when the European Court of Justice, which is actually overruling all the national authorities, they came to the opposite conclusion as the Swedish authority. So this was a cabbage in my garden this summer on July 24. Then it was not a GMO, and the day after it was a GMO. Probably. Because our authorities, they still don't like this. They have not told me to take this away. So this is, now it died a few weeks ago because we had frost up in Umeå. But still, so there is, what I'm saying here is that this was for us scientists or for breeders, it was an extremely unfavorable decision. And because there are plants out there, you may actually want plants with increased vitamin content, resistance to whatever things, and so this may what we may want in agriculture. All of these ones, I tell them, they all exist. They have been around for 10 years, but they are GMOs, so they have not been able to use in agriculture. So you have never seen them. Now we were hoping as a scientific community that suddenly we can redo all these things using CRISPR. So this is what my colleagues throughout the world is now doing. They are reinventing the wheel. It's a brand perfect new technique, but what we are still doing, that is just reinventing what we did, hoping that we can use them. The sad thing is now they will be used outside of EU. And this creates an impossible situation for international trade, because things that are grown just like anything in the US, in Brazil, in Japan, and so on, when they should be imported to Europe, they should be labeled as GMOs according to our laws here. And they cannot be traced, and they have not been traced before. So what this mean in reality is actually impossible to predict. So this, at least for me, put some hope into this and say that perhaps we never had the better case than now to say that we need to totally revise the GMO legislation, because this doesn't make sense. This is the consequence of an inadequate and sort of outdated regulation of these kind of techniques, which should change. So with that, thank you.
Definitely. Thank you so much, Dr. Jonsson. We have time for exactly one question, in case anyone has it. And if not, I'm going to keep you moving on. Yes.
Yeah, I have a question. Thanks for laying it out so entertaining. Namely, how does the field, and maybe journalism, so maybe this is a question for Jennifer Kahn too, handle the public attention and the responsibility that comes for the public discourse? So I'm thinking here, for example, about the was a Schaefer-Wong study 2017 and Nature Methods paper that purported to show safety concerns about CRISPR by showing sort of off-target effects. And so the study was eventually withdrawn because of major problems in the study design, where they didn't properly account for natural mutations and things like that. But not before a giant media freakout, biotech companies, stock crashing, and sort of, I'm thinking back here to sort of the problem we have with vaccine like deniers, right? Where a single problematic study, it does not matter that it is retracted, right? The original study that drew any analogy between autism and vaccines was also retracted, and it's a faulty study, right? But it's now out there, and it's being used by anti-GMO activists and so on. So if apparently big journals cannot protect us. I mean, this was not some silly journal with Nature Methods, right? It doesn't get bigger than that. If the university press offices don't protect the public discourse, if the media certainly doesn't, because they're jumping on this hype like everybody and reporting on preliminary study results within minutes, then the responsibility falls to the scientist, does it not?
Yeah, then people like me have to go around and talk to people like you like this. That's the only way to do it. I should say, when you talk about off-target effects in plants, that's not an issue. All the things I told you in the beginning of plant breeding is non-target effects. So what you are if you're creating non-target effects using CRISPR in plants. That's exactly the same things that is not regulated. So for us, it's not, but it's maybe a problem in medicine, I can agree, but really not for us.
All right, and to round out the evening, we have Dr. Paul Hudson talking about CRISPR to make biofuels using just sunlight and carbon dioxide.
Thank you. I'm trying to figure out I wasn't paying attention earlier when Stefan had to do this.
Just close it down on your presentation as well.
Oh, thank you. Yes. Good, you can't see what I'm doing on the screen. It's embarrassing that I keep clicking the same thing over and I think I got it now. Now I have to just make it slideshow. Great, all right. So I would also, am I heard okay? Good energy. I would also like to thank the organizers for inviting me to give this presentation. I will talk about CRISPR-Cas that has been described so far in some detail, but in a different application, and that is mostly, well, it is totally biofuels. A little bit of biology in there as well. So the title of my talk is a bit ambitious, and I've heard that you're not supposed to start with a question in the title because the answer is always no. So if you read articles or in journalism, when it's a question in the title, the answer is always no. So I'm not going to reveal the answer just yet, but it's about biofuels, a new way to make biofuels. Okay, great. So I will begin with a little bit of motivation. Of course, everyone here knows about biofuels. We've heard about biofuels probably for most of your life, and you're wondering, what role does biofuels play, really? And in Sweden, biofuels play quite a big role in industry. So this is the energy distribution provided in Sweden, divided into industry on one side and transport on the other side. And you can see that in industry in Sweden, we're quite good using renewable fuels. Renewable fuels, in this case hydroelectric and nuclear, really help us out, but also biofuels. In transport, it's kind of our dirty little secret in Sweden. Sweden is thought to be a very progressive country. They put a lot of research funding into alternative energy. I know that. We think green, we take the train, but it is quite like Rome, how Rome was a very messy city for the Romans, but when they colonized the world, all their streets were straight, but back home it was quite dirty. This is kind of how it is in Sweden, right? So the problem here is that Sweden is importing a lot of oil. So I don't know how many of you understand the magnitude here. 10 billion liters of oil per year just for transportation. So biofuels can take some of that, relieve some of that oil dependence. Of course, electricity will have a role with cars, but biofuels will have a role as well because the technology is quite developed, and you can also enter the aviation sector, which is something that is happening right now for biofuels. I think, yes, okay. So my research is focused on a new way to get biofuels, and I like to contrast by starting with the kind of state-of-the-art for getting biofuels, and that is to use fermentation of sugar that is derived from either wood or wheat. There are other biomass sources as well these days. But in any case, there is a processing step from the natural carbon source. It starts with CO2, of course, and sunlight. There's a natural carbon source that has to be converted to sugar first. The sugar is then fermented into a biofuel. And this can be used, you can use a natural yeast that ferments to ethanol. You can now use genetically modified yeasts, for example, that can make butanol, and even that's butanol here, 4 carbons, or even longer chain, more exotic fuels. But these are now combusted, and the CO2 can then be recycled. And this is not an infinite loop. It does have an energy input, and that is the sun. So the sun is the ultimate source of all our energy, of course. So my research group works on a way that simplifies this process. It shortens the path. And that is that we use a photosynthetic bacteria called a cyanobacteria. And it does a very neat thing, is that it fixes CO2 and sunlight inside a single cell. And with some genetic modifications, so you can see where this talk is going, with some genetic modifications, this cyanobacteria can be coerced into producing biofuels. Right, so this is what we work on in my group. At SciLife Lab, I'm situated there at SciLife Lab, we grow them in small scale, in flasks, on plates. We grow them in a little more advanced small bioreactors here, and other places they're growing them outdoors in very controlled So how is this going for us? Right? I've been working on this for some years now. In the state of the art right now, I can summarize quite succinctly, is that they do produce biofuels, cyanobacteria, with some genetic modification, but they do it with low efficiency. And in this slide, I'm showing the path of sunlight energy as it is used by the cyanobacteria. And you can see that if we normalize to the sunlight being 100% incoming, only half of that is visible to the cells. So already half of the available sunlight is gone. They don't see it. They're green, right? Green light's not absorbed, right? That's why they're green. So half of it's already gone. There are groups working on improving that, but not me. Then we have another drop in efficiency, and that is about a quarter of that is gone because of reactions that happen in the photosystems that Stefan was talking about. I will come back to that at the end, but this is also quite not really available for us. It's kind of like thermodynamics are preventing us from accessing that. So the heart of the matter is the best we can do is this 12%. That's kind of like the theoretical yield right now. And the best we've achieved in my lab is 1.5%. And that's under very well-controlled conditions. Sorry, was my microphone a little, it was a little off. Let's see, I have I think a respectable goal, this is really not, I guess I have to hold it down. Yeah, okay. Sorry, I have a certain flow, and now the flow is interrupted because I don't know how long to push this silly button. In any case, our goal is to improve this, to engineer up to 10% conversion. And to give you an idea of what efficiencies we're talking about, I've plotted here the area that you would need to cultivate current crops to reach this 10 billion liters that we import every year, right? I told you Sweden imported 10 billion liters of oil. Now, if you're going to provide that energy using bioethanol from corn, you would need a cornfield this big. Okay, it seems pretty big, but America is very big, right? With very few people around, and that's where, or you know, that's kind of how we're importing it, right? If you're going to do biodiesel using rapsolia, oh, switching horses midstream. I feel like this is also warned against by Abraham Lincoln. In any case, if you're going to grow biodiesel, make biodiesel with raps, you need a field this big. Now, our cyanobacteria that we grow in the lab, at 1% conversion, you need something only this big. So it tells you that the conversion efficiencies of these are much lower than what we achieve already with cyanobacteria. If we reach 10%, our field gets that small. Now, I put these up in Pitio. It looks silly, maybe. Why would we ever grow in Pitio? But there's an interesting fact about Pitio, and that is that it gets a lot of light in the summer, and the temperatures don't get too hot. So that's actually quite a benefit, is that the cells, they don't stress out too much. Originally, people put the cyanobacteria in the desert, and they thought, lots of sun in the desert, this is a great idea, genius. But then the cells died in the summer because it got too hot in Arizona. So this is actually a good balance, I think. So the CRISPR-Cas technology, it just entered our field of genetic engineering of cyanobacteria, and it is really it's a enabling technology, they call it. So I got this slide from Jens Nielsen at Chalmers, and he works with yeast metabolic engineering, and he presents the existing technologies taking us from a proof of principle strain, so a strain that makes a small amount of biofuel. Existing technologies can take us to our final strain. We have high titers, rates, and yields, everything we need to make a biofuel from a cell. But it takes a long time, 100 person years, they estimate. And that's with an established host like yeast. For cyanobacteria, this will take probably longer. Now, with novel technologies, such as the CRISPR-Cas9 technology, we think that this time can be shortened considerably. There's already an example that was just brought to my attention from a company out in California. They're funded by Exxon, and they have used the CRISPR-Cas technology to increase oil from an algae, so not a cyanobacteria, but an algae. And this was a paper published last year. And the interesting thing about this paper, I just want to highlight, lipid production is doubled by decreasing expression of a single transcriptional regulator, one protein in this algae. Changed one protein, just the level of one protein, and it doubled the amount of oil, just like that. So why could they never find this before? And the reason is because it was very difficult to genetically engineer algae targeted in a targeted way. And now with CRISPR-Cas, they can do it. So they're discovering all types of And another thing about this is that there's 20 people on this paper. So it takes an army to do this type of work, actually. And now Exxon, this company is from Craig Venter. He sequenced the human genome. He's now working on algae. And Exxon has now thought, with this type of improvement, we can scale up 10,000 barrels a day in 10 years. 10,000 barrels a day sounds like a lot. That's 1% of what Sweden uses, actually. But it's a start. So when I think about where our work is going to go, getting to that 10% yield, I think there's three areas. One is that CRISPR-Cas9 can help us with. One is optimizing the biosynthesis pathway, so the enzymes that synthesize the fuel and removing competing enzymes in the cell that kind of deplete from that pathway. A second is to find about native regulation in the cell, and this is kind of what I hinted at with the algae guys, is that they found that a transcriptional regulator means it controls many genes. They identified that, and they were able to change it. Now, we want to do the same type of thing, and I'll show you how CRISPR-Cas is helping with that. And then at the end, I can talk a little bit about new industrial phenotypes that we're interested in. 10 minutes, sorry. Yeah, okay. So CRISPR-Cas9 helps us in a big way because we can now quickly do genetic engineering, so like gene editing that Emmanuel and Stefan were talking about. So in my lab, we use metabolic models. So we try to simulate the metabolism on the computer. So when we have a starting strain, like our kind of weak biofuel producing strain, he makes a little bit of biofuel. And we want to create a monster strain, something that makes much more so we can really save the planet. Right? We simulate the metabolism in the computer, and we ask the computer to tell us which mutation should we make? And the computer can do something like this. It can simulate the metabolism where you have the phenotype here of the cell, and the cell can take its incoming carbon, and it can either grow or it can make biofuel. Right? There's only so much carbon coming in. It can do one or the other. Right? The cell is allowed to exist anywhere in this green space, meaning I can exist here where I make a little bit of growth and I make a decent amount of biofuel. Okay. Cells tend to hang out down here, though, sadly. A bacterial cell has evolved to grow. That is its primary motivation in life. It does not evolve to make biofuel, so they tend to hang out down here. So what we want to do is we want to reshape the metabolism, like shape the room by making targeted knockouts so that only this area is available for the cell, right? So if he tries his old tricks and tries to grow, he's actually going to have to make biofuel, right? This was the dream 2 years ago or so, yeah? 3 years ago. This is what we were doing. I have a very talented grad student working on these models. The knockout list to make the metabolism look like this, however, is very long. Many, many knockouts, 10, 12, 15 knockouts you have to do. That's not feasible with the old technology. It's barely feasible with the new technology, I think. But we can begin to take the steps. It's completely daunting to make 15 knockouts in a single strain without CRISPR-Cas. So this is one way it's helping us go forward. We have not made all these knockouts, for full disclosure, a couple of them. The tool that we've been using the most, actually, in our lab is what's called CRISPR interference. And Emmanuel, she touched on this just briefly. And this is a variant of CRISPR-Cas9 technology that allows partial repression of genes. So not the whole knockout, just squeezing the gene down a little bit, a knob, as Jennifer called it, right, to control the expression. And we think this is a very, very wonderful tool. And the way it works is that you inactivate those DNA-cutting parts of the Cas9 enzyme. So the Cas9 enzyme, it still is guided to the target DNA. It sits down, but it doesn't cut anything. It just sits there. And the transcription of the gene is blocked, right? So it really just literally blocks the transcription. Now, we were the first group to do this in cyanobacteria, to port the CRISPR-Cas to cyanobacteria. And we did it first with dCas9. And we showed in this paper that the dCas9, after we optimized it, it could be easily controlled, meaning we can turn it on. So here's a gene that's green, GFP. It glows green. We can watch it with our eyes. We turn the repression on, and the GFP goes off. We remove the inducer molecule, so we turn the dCas9 off, and GFP comes back on. So we can control it. That's very important for basic research. It can also be multiplex. I'm not showing the data here, but we can do up to 6, 7 genes we can knock down at the same time. So this really accelerated our basic research. Now we'll show you 2 examples of how we're using CRISPR interference, or as we call it, CRISPR-I. So one of them is to use CRISPR-I to block growth. So instead of doing all this crazy restricting the metabolism with knockouts, we can just simply tone down one of the key enzymes responsible for growth, right? So this is what we call a metabolic switch from growth to making biofuel. So we've put this Cas9 and it's targeting now, it's going to block expression of an enzyme or a gene called GLTA. That's citrate synthase for those of you who care about that. And here I'm showing cell density, so the growth of our cyanobacteria. And you can see that the normal strain grows quite well over some days, but the strain that we have repressed, this GLTA, it does not grow so well. Okay, it doesn't grow well, but does it make more butanol? Yes, it makes a lot more butanol than the wild type, or than the starting strain here. So it makes quite a bit more. Another benefit is that the cultures, they don't get too dense. This is something that's bad for cyanobacteria. If the cell culture gets too dense, light cannot penetrate to the guys in the back, right? The cells in the front, they take up all the light. They take up too much light, as Stefan said. They get hot, they get stressed. But the ones in the back get nothing. So this is a way to control the density of the culture. And we have quite good conversion over the course of the cultivation. So our butanol story so far, when we started in 2013, our strain made very little butanol, right? I mean, barely detectable. Over the past couple years, using our genetic knockouts and now using the metabolic switch, we are up to now, well, this is a year old, we're up now to about 1.5% conversion efficiency. That's under laboratory conditions, however, to be clear, very nice, pleasant conditions for the cell. We will see how we continue. So this is just a picture of how we've modified the cell. So we've added new genes, we've removed genes, during the course of our work. So in the last few slides then, I want to talk about how we're trying to take this last step, going from 1.2 to our goal. How do we get there? The last step is probably the hardest, actually. A lot of the low-hanging fruit is gone, turns out. Well, we're using CRISPR-I. We're going to keep going with this really cool tool. And so we're using what's called a CRISPR-I library. So in this particular case, we synthesize chemically, we synthesize these guide RNAs that Emmanuel talked about so much. Now we use the design rules that her group has published. So we follow the advice. We can synthesize sgRNAs targeting all genes. So every gene in the genome, thousands of genes, we're just going to repress them all, right? So each cell will take up one sgRNA. So each cell will have one gene knocked down, right? So there's about 15,000 members or so of this library. And then we're going to make this library, all these mutants, we're going to make them compete with each other. We're going to fish out the ones that are best. Yeah? So we're going to create the library, and then we're going to screen the library. We're going to look through all the mutants and find the one that is behaving the best. So in this particular case, we've grown the mutants at high light conditions, so really high light, and we found mutants that actually grow faster than the wild type. And when we looked at the sequence of these, when we started to investigate which mutants were there, which ones were actually present at the most at the end, so the ones that actually thrived in this environment, we found that they were transcription factors. Right? One of them was actually a known transcription factor, which altered the ratio of PS2 and PS1 in the thylakoid membrane. So this is a mutant that we never would have guessed ourselves, actually. I'm not clever enough to think, oh, we have to adjust the ratio of these two proteins. I'm not that clever to find a fast-growing mutant. But we found it because we randomized the mutants, and we did that using the CRISPR-I technology. We can also use more advanced, so not just growing them and seeing who survives the best. We collaborate with groups at KTH that are sorting the library using quite advanced microfluidics technology. So we are also doing to see if they're secreting more of the product, such as butanol or other chemicals. So it's not just growth. We are a little more advanced. So I've talked a little bit about how to do the gene editing. with the models and guiding the gene editing, and also about discovering new regulators. And now in the third final act, I will just mention one way we're trying to improve the processing capability of the cyanobacteria. So I had a very talented master's student named Astrid who looked into the economics of growing cyanobacteria up in Piteå, as I said. And she did some good simulations, and we had a lot of assumptions in these simulations. But the conclusion of these simulations is that the greenhouse gas emissions of making cyanobacteria produce butanol in general are higher than making ethanol using current technology. So some types of ethanol, such as from sugarcane, it's quite bad for the environment. The greenhouse gas emissions are quite high. Other types are kind of low. We simulated different cultivation technologies using cyanobacteria, and we found in general they are higher than the ethanol. Kind of in parallel, we found the energy demand to make cyanofuels right now is also higher than it is to make ethanol. So right now, this is not good. We would not go into the field and do this now. This tells us it's not there yet. But it also gives us a hint about why it's not there. Why is the technology not there? And it turns out that when we break down the factors for why the greenhouse gas emissions are so high, one of them is this cultivation process. And that has to do with mixing. It has to do with a lot of water that are needed to cultivate these strains. So we start thinking, how can we reduce that? And one of the ideas is to make the strain, and this is my final slide, one of the ideas is to make the strain immobilizable on a surface. Now, most cyanobacteria, sadly, they cannot be immobilized on the surface. We have an idea to immobilize on the surface. The cells secrete their biofuel out of their little green bodies, and we remove the cells to purify the fuel, right? So it's low water, and the cells are durable. They can be reused. So I had a grad student in my group that used gene fusions to attach, to make a strain that is sticky, essentially, that can stick to a surface. And her idea was to fuse a sticky protein to different parts of the cell surface. So these are 3 different proteins that she's drawn in a schematic. Obviously, this is not a real picture of the cell membrane. She's drawn them in here, and she attached this sticky protein, a genetic fusion, so that the sticky protein is on, like, part of these. And then she took pictures. And the sticky protein in these pictures is in green. So one of them she tried, not very good, right? The sticky protein is not on the cell surface, right? One of them, the sticky protein is all around the cell surface, but it's not sticking out very far. And on the last one, when she fused it to this appendage, this pili that sticks out from the cell surface, you can see lots of green protein far away from the cells, which are in red. So this is truly hanging off like a string, right? And we think that this cell will be very sticky, right? That it can stick to surfaces. But the point I'm trying to make is that this took her a long time. She had to test many different gene fusions in the lab. CRISPR-Cas had not come to cyanobacteria, sadly, or at least not to our lab at that time. Nowadays, this can go much faster. Nowadays, just 1 year ago, really. Yeah. Okay. So I will just, in the end, just thank you for having me again, but also my wonderful research group at KTH and SciLife Lab. And we have funding mostly from Swedish universities, but also some private funding that's working with on this. And I thank you for your attention.
All right, we are close to on time. We can probably take at this point 1 question or 2 if you keep them very brief, and then we'll wrap up for the night. Are there any final questions for Paul or I guess any of the panelists? Yeah, any of the speakers.
I can ask a question that I realized I didn't answer, and that was, is it feasible with CRISPR-Cas9? And I think, I don't know, I think
Tell
the
me,
answer
Paul,
would be
is it
yes.
feasible?
Yes, I think it is feasible. I think we can reach that limit. I do think it is feasible. Yeah. Yeah.
All right. Yay.
This is not going to be made public or anything, is my statement. No. I'm not on the record, am I? Okay.
Well, then it looks like we're wrapping up for the evening. So thanks to all the speakers. Thanks very much to IVA for hosting this. And I guess Stefan will take us out with a few words.
I will not spend much time up here, but I think all of you has understood that the title, Genome Editing, and the enabling tool that Emmanuel Charpentier has provided to all of us. And then Paul will if we get some more years, he will save us in terms of energy. Stefan will change the EU legislation and get crops that we could benefit from. And many researchers also in this room use CRISPR technology for medical applications. So I really think it's an absolutely fantastic thing. I want to thank all the speakers in a very small way, giving them a memory for the night. So I start with Emmanuel. And of course, Jennifer. Thank you very much. And of course, I thank all of you who came here listening. Unfortunately, there wasn't that many questions, time for that many questions, but still we have learned a lot. So thank you all. And now, for those who have registered for dinner, it's time to slowly move up. Thank you very much. Bye.